Module 7, the nervous system. Neuroglia. The second type of nervous system cells are neuroglia, sometimes called glial cells. The word neuroglia actually means neural glue, and neuroglia are indeed the connective glue of the nervous system. I'll be spending a lot more time on neurons in this course, but it's still important for us to talk about neuroglia because most of the nervous system is made up of these cell types. Neuroglia are small in comparison to most neurons. Now, you already learned that neurons cannot undergo mitosis after infancy, but some people get brain tumor growths. A tumor is a location where there is uncontrolled mitosis. Well, how can there be uncontrolled mitosis in the brain when the neurons in the brain cannot undergo mitosis? Well, most brain tumors are from mitosis of neuroglia because these cells can reproduce. There are 6 types of neuroglia cells, and each has a different job. The first type of cell is an oligodendrocyte. These are found in the central nervous system, and they have 2 jobs. One is to connect neurons together. Here you can see how one oligodendrocyte is connecting many axons together. And because the oligodendrocyte surrounds the axons, it provides insulation for them as well. In the peripheral nervous system, Schwann cells also insulate, but they only wrap around part of a single axon. Microglia help to fight off infections using phagocytosis. Remember, phagocytosis is when a cell engulfs another cell and breaks it down. Microglia do this throughout the brain and spinal cord. Thankfully, we rarely get infections in the brain and spinal cord, but there is a method of protection if we ever do. Astrocytes are responsible for creating the blood-brain barrier. It's a physical separation between the blood and our brain, and you will learn more about this amazing design later on. For now, know that in your circulatory system you have very tiny blood vessels called capillaries, with walls so thin that they can absorb waste products that cells produce. They can also release nutrients carried by the blood out into the surrounding tissue. But some of those materials are deadly to neurons, so astrocytes have processes that surround the capillaries so that a barrier is formed. The astrocytes, along with special cells in the capillaries, work together to take out of the blood only what the neurons need. In this lab, we're going to get a close-up look at some neurons in the brain. We're going to also be able to identify how they are more distinguishable from neuroglia. So we're going to take a look at the human cerebrum slide. Let's put that underneath the microscope at 40 power magnification. And focus. Go ahead and center that just in the center of the pinkish region. And then move it up to 100 power magnification. And focus. Now at this point, you're going to see lots of white holes or white openings. I want you to focus on some of those and look for some that have a cell in the center of that white circular area. Center that cell in the middle of your microscope and go ahead and move it up 400 magnification. And focus. What you're looking at right now is a neuron. That's the cell that's inside that white section. And you can identify that it's kind of irregularly shaped. Take a look at a couple different cells like this in your sample and make an illustration of those in your lab notebook. You're going to notice that they're shaped a little bit differently from one another. They're varied in their structure, and they have extensions. Those extensions are processes, and you can begin to see just the beginnings of those processes based on the cross-section that you're looking at under the microscope. So they should look like sharp points extending out of the neurons. Most of these neurons are multipolar. And that's because the neurons in the brain are association neurons, and they have to communicate with lots of different cells, depending on what the signals are that they receive. So make those illustrations, and once you've done that, go back and take another look at the pink material surrounding these circles you were looking at. That pink material is made up of fibrous material, and those fibers are dendrites and axons. That's a lot of dendrites and axons that you're looking at compared to the number of neurons that you're seeing. There's a lot of interconnection between the cells in the brain so that they can communicate to one another. It's really fascinating to see the combination of those as you're looking at it under the microscope. So once you've done that, go ahead and clean everything up and put it away. You probably have figured out that your brain has several protective systems in place. and the astrocytes that support the blood-brain barrier play a major role in that protection. But though the barrier prevents many toxic materials from getting into the brain, it's unable to prevent substances such as illegal drugs. These chemicals are able to penetrate that barrier and can damage brain cells. Alcohol is also able to pass through the barrier, and many studies have shown that it isn't good for brain tissue. Another type of neuroglial cells are ependymal cells, and there are two types of ependymal cells. Some have no cilia, and their job is to secrete cerebrospinal fluid, which engulfs the brain and spinal cord tissue to give it a protective cushion. Ciliated ependymal cells move the cerebrospinal fluid around.